Hi everyone. Am I audible?
Yes, ma'am.
Yeah, thank you. I'm going to wait for 3 minutes and then I'll start sharing my screen. In the meanwhile, if you guys have any questions, please do ask. And they've also sent the revised assignment solution for week 4. There were some mistakes in two questions. The answers provided was wrong, and the marks were also wrongly assigned. So they've corrected that. So they have also sent the revised solution, and they have mentioned that the marks will also be corrected and it will reflect on your NPTEL portal. So please note the correct answer now so that you remember that when you're taking the final exam. I remember a couple of people had asked that question during the class as well. So just make a note of that. I'm just gonna wait for another 2 minutes maybe before I start sharing the screen. All right, let me share my screen and start presenting. And in case if someone joins, I'll admit them. Is my screen visible?
Yes, ma'am.
Yeah, thank you. So today's class is also going to be a continuation of microbial ecology methods. We saw a couple of them last time, how to taxonomically classify them and also to classify them based on their characteristics. So today is just going to be an extension of different methods, including the various sequencing technologies that are available to us, various microbial and molecular biology methods that are available to us for classifying the microorganisms. So this session is going to be little intensive on molecular biology. So if you're not understanding something, please do stop me in between so that I can explain it in detail. And especially if someone is not from biotechnology background and does not understand much about some of these concepts, please do stop me. So this is something that we already saw in the previous session itself, that not all microbes can be cultured in the lab, and this is what results in the great plate anomaly, that if we take a plate of microbes collected from an environmental sample, only like 0.01 to 1% of the bacteria in the environmental sample will actually be culturable or can be grown under laboratory conditions in plates or culture tubes and so on. And we also saw that even for bacteria that cannot be cultured, 16S rRNA kind of genes can be used to identify what microbe it is, what bacteria it is. And we also saw a list of features which make 16S rRNA superior gene for identifying bacteria. how it is ubiquitous, present in all the organisms, how it is highly conserved, how it is a smaller size gene which can be easily sequenced, and how the large pool of data available in the database makes it easy for comparison and building a phylogenetic tree. There are also genes like metabolic genes. For instance, the genes that are responsible for conversion of ammonia to nitrate are present in certain nitrifying bacteria. So these kind of metabolic genes, for instance, the ammonia to nitrate gene is called as ammonia monoxygenase. That's the enzyme. The gene that is responsible for producing this enzyme is specifically found in this nitrifying bacteria. So in cases where such kind of unique genes are present in a particular bacteria, they can also be sequenced in order to classify them or to characterize them. So in addition to 16s rRNA type of ubiquitous genes, we can also use these type of metabolic genes that are specific to a certain bacterial group or a community. Yeah, these are some of the molecular biology methods. So we have the gene. How are we going to sequence it out of the pool of genome or genomic DNA that we have? We will look at one method after the other. So the most commonly used method is a method where we use restriction digestion. So restriction digestion is carried out using restriction enzymes. These are enzymes, mostly enzymes of bacterial origin, that have the ability to cleave DNA at specific sites or sequences that they recognize. So why do restriction enzymes are available, like why do bacteria or microbes have these restriction enzymes? So this is again to do with their immunity. Just like how our human body has different ways to tackle microbial infections and so on using antibodies, etc., this kind of low level organisms that has very small genomic size, instead of having an elaborate antibody based response against any infection, they have sorry about that. Yeah, they have restriction enzymes which will cleave the DNA of any foreign microbe that is infecting it. So let's say, for example, bacteria get infected by bacteriophages. So there are these sequences in that are recognized by these bacteria that can be cleared by these restriction enzymes, and these sequences are usually sequences from the viruses or the organism that infects the bacteria itself. So these restriction enzymes will recognize that motif or the DNA sequence. It will attach to that region and it will create these cuts that will cleave the DNA of the organism, thereby rendering it inactive or stops it from reproducing further. So this is the reason why bacteria have restriction enzymes, and we have used this for our molecular biology techniques. This is one of the major milestones in molecular biology where we are now able to assess a large pool of DNA, let it be ribosomal DNA or genomic DNA. And when we cut it into smaller pieces with restriction enzymes, we will now be able to assess the pattern of the cut sequences. So we know that what are all the regions at which the restriction enzymes will cleave. So we will be able to assess what might be the DNA sequence at that particular region where it is cleaving. And after that, when we run it on a gel, depending on the weight of each of these cut smaller portions, we'll be able to compare and figure out what would be the possible sequence of the whole DNA that we are looking at. So based on that, we'll be able to reconstruct the sequence of DNA from the microbial community. So this is how the patterns are compared using a gel electrophoresis. So what is a gel electrophoresis? Here it's an horizontal gel electrophoresis using agarose. Agarose crystallizes to form pores, and a tight network along with pores. Depending on the percentage of agarose used, the pore size will change, and when the DNA passes through the agarose, depending on the molecular weight of the DNA molecule, which is in turn decided by the sequence of the DNA molecule that we are running on the gel, they will separate at different places. So at these different, so the higher molecular weight ones usually stay on top, and the lower size ones run towards the bottom. And by comparing these different sequences, we will be able to get an idea about what nucleotide sequence could be present in each of these sequences. So that's how an agarose electrophoresis works. So what are all the different methods through which we can assess an environmental sample? Uh, there are two different types. One is culture dependent. So the 0.1 to 1% of the microbial community which can be cultured can be assessed through some of these steps. High throughput, there are several high throughput methods. Traditional methods involve culturing them on plates and so on, or enrichment based culturing, and etc. After that, we have to isolate the gene of interest and then screen, and then even amplify it by using transfection techniques. So there's transformation followed by transfection, and then we can do expression of that particular gene and then study the character of the gene. For instance, gene like this ammonia monoxygenase that I had mentioned. So if we know that this particular bacteria expresses this, and if we are able to culture this on culture plates, we can isolate this particular gene and then put it through these different culturing methods, and we can express it and finally see how it converts ammonia to nitrogen and characterize that particular gene. So this is for bacteria or microbes that can be cultured and can be expressed under laboratory conditions. But what about the ones that cannot be cultured? That is where all the metagenomic techniques come into picture. We just collect the whole sample, do a whole cell extraction, do a whole genomic DNA extraction, which is the metagenomic DNA extraction. Right. All right. Yeah, which is we can now amplify certain genes of interest like 16s rRNA, or amplify all the genes, construct a whole library, and then do functional screening for the genes of interest, which again will give us an idea about their characteristics. It can be sequenced, like a whole genome sequencing can also be done, and functional annotation can be done based on the gene sequences, and from the gene sequences expression pattern can be analyzed. So these are the different ways through which we can look at the metagenome, or the genome of the whole microbial community from a given environmental sample. So what happens in a metagenomic analysis? We had seen this in a couple of classes before as well. We collect the whole environmental sample, do cell extraction, do DNA extraction, and then fragment it. This fragmentation can be done in a non-specific way using enzymes that just cleave DNA, like DNases that just cleave DNA without any specific pattern, or it can be done using restriction enzymes which cleave DNA at particular sites. And followed by this, there are several techniques we can use for sequencing these fragments. So we'll see how Sanger sequencing is done, and also how the next generation sequencing is done in a couple of slides. But we do DNA sequencing, and once we have the sequences, we classify the reads based on the sequences, the DNA sequences that we get. We can either do a taxonomic profiling to understand the phylogenetic positioning of these different microbes, look at which microbes belong to which communities and so on, look at the virulence factors to see how the infectious they can be, the other infection profiles can be assessed, and also the functional profiling of overall what all genes they possess, how what all are their metabolic functions and so on. So that kind of profiling can also be done based on the metagenomic sequences that we have. Another very interesting technique that is helpful in understanding phylotypes. So what are phylotypes? Uh, this is like group of microbes that are very similar in certain characteristics or certain genes. They share certain genes and can be classified together in a phylogenetic tree. These are called phylotypes. So to differentiate different phylotypes together, we can use this method called denaturing gradient gel electrophoresis. So how does that work? Is that let's look at the nucleotide sequences. So these are how the G and C. So in DNA, the guanine binds to cytosine, and adenine binds to uracil or thymine. Uracil in the case of, so here we can see that uracil or thymine. So here we can see that there are two hydrogen bonds between A and U, whereas in case of G and C, there are three hydrogen bonds. So to break the three hydrogen bonds, more energy is required as compared to AU or AT based binding. So this particular principle is taken into account, and this is what is used in case of denaturing gel gradient gel electrophoresis, where like I mentioned, since GNC has three hydrogen bonds, more energy is required to break these three hydrogen bonds. So here in this case, as the GC content increases, that is, if there's more GC sequence in our sequence of interest, the melting point or the melting temperature of that particular sequence will be very high. So as you can see, at 100%, the TM increases as high as 100 degrees Celsius and so on. So this idea is what is taken into account in a denaturing gradient gel electrophoresis to separate phylotypes. Sorry. Yes. So here we cast a gel. So instead of the acrylamide gel, instead of the agarose gel that I had mentioned in the previous electrophoresis, here it is a polyacrylamide urea formamide gel. So this is what creates this denaturing concentration. So at these different regions of the gel, the concentration of urea is different. So it changes from a lower concentration and goes on to increase to a higher concentration. So when we load samples, the DNA starts migrating towards the positive electrode, and it moves through this denaturing concentration. So depending on the GC content, just like TM, this denaturing condition also changes. So more concentration of urea is required to denature sequences that have more GC content. So as they move through this, the sequences denature or break at these different regions, and we can see these patterns being formed as DNA migrates. So the ones that have lesser GC content will stop in the top, whereas the ones with higher GC content will move further down. So as we can see here in this example, wild type, which does not have any mutation, moves down further, and on various samples we can see that there's alternatively mutant 1 or mutant 2. So this mutant 1 has one nucleotide difference, and hence it is slightly above the mutant, above the wild type, whereas the mutant 2 has more GC content, and it is sorry, that has lesser GC content, and hence we can see that it is on the top, and it has two mutation points. So therefore it is two steps higher than the wild type. So therefore we can differentiate these different phylotypes. So whichever the ones that are here near mutant 2 belong to a particular phylotype, whereas these ones belong to the wild type, and this ones belong to the mutant 1, and so on. So that's how we can use the environmental sample, get the DNA from that, and just by running them on a gel that has denaturing conditions, we'll be able to see how differently they have these different mutations. So we have to amplify them using specific PCR primers before we run them on the gel. We're not running the whole genomic DNA here, we're just running our sequence of interest or gene of interest. But our from in our gene of interest, we'll be able to differentiate phylotypes by using denaturing gradient gel electrophoresis. So that was one method to separate different genes with single or double mutation points. But there are also other techniques similar to this to understand how these mutations happen in these microbes and how they convert them into different strains or different phylotypes. Another very interesting technique is restriction fragment length polymorphism. So this is to assess polymorphism at the sites that are that can be cleaved by these restriction enzymes. So this particular example that they have shown here is for a normal beta globulin gene, beta globulin allele, and a sickle cell anemia beta globulin allele. So in sickle cell anemia, this point mutation results in the globulin genes to change, which is what changes the shape of the RBC cells in comparison to the normal RBC cells, which results in lesser space for the RBCs to carry oxygen and so on. And there's also this whole idea about how this single point mutation gives resistance against malaria and so on and so forth. But here the thing that we are going to look at is how restriction enzymes can be used. So I had mentioned about restriction enzyme in a previous slide as well. So here we take these different restriction enzymes and add them along with our genes of interest. So instead of these globulin allele and sickle cell anemia allele, imagine we have environmental samples and we have amplified them using so here RFLP can be done for the whole genome. We don't have to amplify using PCR first. So this is a technique that we can directly go with the whole metagenomic sequence. So we are just using different combination of different restriction enzymes to cleave the whole genomic DNA. So let's say we are doing this for our microbes of interest, and then we get this kind of a pattern. So restriction enzymes can be used and the cleavage patterns can be compared to get an idea about how the different alleles are there in a particular gene. So here in this particular example, as we can see, the first one is a normal allele, the second one is the mutant allele, and the third one is an heterozygote, where it has one normal allele and one mutant allele, and it's an heterozygote, thereby carrying signatures of both the normal allele and the mutant allele. So here we can see that the bands, the ones that are there in this as well as in this, are present in the heterozygote. Similarly, for our microbial community as well, we can assess the point mutation polymorphisms and classify them depending on which allele they have. And this is further plotted onto a nitrocellulose membrane, and radioactive tagging is used to specifically understand about our genes of interest, because this pattern now is for the whole genomic DNA. From this, if we have to specifically look at our gene of interest, we can radioactively probe it with that specific gene sequences against that specific gene, and then we'll be able to get an idea about what are all the different mutations that are there in our gene of interest, thereby helping us to classify them as different strains or different microbial communities or different phylotypes and so on. So that's how restriction fragment length polymorphism works. So the next one is okay. So far, if there are doubts, please do stop me, or maybe I'll just stop for a bit and just quickly check if there are any questions. So far, is it clear? Are there any questions? Because this is a little heavy on molecular biology, please do clarify if you have any doubts at this point before I go on further.
Ma'am, in the same RFLP,
Yes.
you have written this restriction fragments. So is this method very specific? Like we have to use any enzyme or by other methods? Like,
Yeah, so in RFLP, yeah, so we use specific restriction enzymes. So each restriction enzyme recognizes a particular cleavage site. So if we already know what kind of a sequence we are expecting in our gene of interest, then we can use just one restriction enzyme and cleave it at that site and look at the polymorphisms. But in cases where we do not know where exactly our gene of interest lies or what is the sequence, we'll just cleave the whole genomic DNA with a combination of multiple restriction enzymes. So far, several this kind of restriction enzymes have been isolated from various bacteria like EcoR1 and so on. EcoR1 is a restriction enzyme isolated from E. coli bacteria. So there are many such restriction enzymes, and they can be used in combination to get these varied patterns.
so
Is that
yeah,
okay?
we have to decide on what prior knowledge rather than random. Yes, ma'am.
Yes, yeah. So if we don't have any prior knowledge, we can just use a combination of commonly used restriction enzymes. So we already know like which sequence it recognizes and cleaves. So we can use some of those commonly used ones. But if we already have some prior knowledge based on the data available online, if we are just looking for that particular, let's say for example, we are looking for a nitrifying bacteria in our environmental sample of interest, and if someone has already sequenced some of these genes from the other nitrifying bacteria, we can just use that information to select the restriction enzyme.
Okay, ma'am. Thank you, ma'am. I
Yeah,
got it.
yeah, okay, thank you. Any other questions? Anyone wants to
Like
clarify something before I? Yes, sir.
same questions, the answer is they are comparing this with a library already created. That's what he actually wanted to know.
Which one, sir? They comparing which
Same
with
question
the?
he asked in the previous question. He asked for the same because we are just restricting. We are giving this digesting with the restriction enzymes, and we are studying the pattern.
Yeah.
So we are want to know how it will be. So we are just comparing this patterns with the previous patterns available, like a library. So we'll find out what organism will be there in that.
Yeah, so we can do that, or we can compare within our samples only. So here, if we compare within our samples, we see that this particular sample has these two bands which are absent in the second lane, whereas it is they all the three bands are there in the third lane, which means that this one is a combination of the first and second one, and hence it's an heterozygote. Similarly, in even in our samples of interest, we have a particular community where we see such a combination of bands, then we can see that thus that particular microbe has is an heterozygote carrying two different pairs of alleles. Let's say for the same nitrifying bacteria example, if we get these kind of a pattern, we'll know that there are two different alleles which are responsible for the nitrification, and this one is an heterozygote which carries both the alleles. So we can compare within the samples as well. But if a library is available and if we already have a prior knowledge of how these restriction patterns will look for our gene of interest, that would be even better, so that we can get a clearer idea of how these point mutations are happening in our gene of interest. Is that clear, sir?
Yes, yes, yes. Yes.
Yeah.
No, yeah. Yes.
Okay. Thank you. So I'll just share the screen again, and yeah, the screen's still visible, right?
Yes, ma'am.
Yeah. Okay. Thank you. So that's how RFLP works. So another technique is AFLP. So here we do not use. So here we actually, before AFLP, I'll probably talk about RAPD. So RAPD is a method where it means random amplification of polymorphic DNA. So here again we are looking at polymorphisms like single site polymorphisms, but here we do not use restriction enzymes, but we rather use random primers and sequence random regions. So these are microbial samples collected from site A and site B, and we are using random DNA primers that can attach to these different regions in the DNA, and we are sequencing all of them, and we are amplifying them using PCR and running on a gel. Based on the gel pattern, we'll be able to figure out whether a particular gene that we amplified is polymorphic or monomorphic. So what is monomorphic? So here this particular band is present in both the site A microbe and the site B microbe, which means that this particular band of interest or this particular gene of interest is common to both site A and site B. Whereas this particular band is present in A and absent in B. Similarly, this particular band is there in B but absent in A, which shows that this gene is polymorphic, and it shows different sequences or different patterns from different regions. So this microbes from this region has this allele, whereas the microbes from this region has a different allele. So therefore, from this kind of information, we'll be able to get an idea about which polymorphic allele is present in which of the two sites that we are looking at, or which of the two environmental regions we are looking at. So that is how random amplification of polymorphic DNA is done. So this completely relies on using random primers that are designed to attach to random regions in site A and site B, and we look at the electrophoretic pattern. So let me go back. So AFLP is actually a combination of both of these methods, where we use restriction enzymes as well as primers to amplify. But instead of having like this lot of data where we just look at the data at the end through electrophoresis, in AFLP we use restriction enzymes. Like I mentioned, EcoR1 is a restriction enzyme, TAC1 is a restriction enzyme. We cleave them into smaller regions and give them these TAC1 based adapters or EcoR1 based adapters. Because we cleaved them using this particular restriction enzyme, we will know that what nucleotide sequences will be there in these ends. So because they cleave like this, they leave these staggered ends, and we know what will be there in this end. So we can now add primers that can be ligated to these staggered ends, and these adapters are created. And now because we have this adapter, we can use primers specific to this adapter with a single selective nucleotide of interest. So this completely relies on our prior information of whatever polymorphic allele that we are looking at. So if you're looking for a specific polymorphic allele in our community of interest, let's say we know that this allele of nitrifying gene is better at converting ammonia to nitrate, and we want to see whether microbes isolated from this environmental site as that particular gene of interest or that particular allele of interest, then we can use a primer that is designed specifically to look at that single nucleotide or two or three selective nucleotide regions, and we can amplify that using PCR and later on look at the gel electrophoresis profile. So if we do amplify it and if we get a single band, then we can be sure that this particular single nucleotide polymorphism is present in this region as well. And further, that can be used for characterization, and we can use that microbial community for our application purposes. So that the AFLP method actually gives us the advantage of using both these methods of using PCR based amplification and restriction enzyme digestion combined together to get the final gene information about the final gene of interest. So that's how AFLP works. And so this table gives a combined comparison of these different techniques: RFLP, RAPD, and AFLP. RFLP is the just restriction enzyme based mechanism, RAPD is the amplification based one, and AFLP is a combination of both. And as we can see here, this needs restriction enzyme, this does not, and this one needs. Just a second. All right. me share again. Yeah, so it require RFLP needs large quantities of DNA because if we are cleaving it with multiple restriction enzymes, we need large quantities of DNA to start with. Whereas since RAPD is amplification based and anyway genes are going to get amplified, we do not need large quantities of DNA, and same principle applies for this as well. So as you can see, AFLP combines the positive sides of both the processes, and it has the positive sides of both the techniques together. So we don't have to use any specific restriction enzyme. Like I said, we can use a pool of commonly used restriction enzyme and get the whole idea about different restriction patterns. But here in case of AFLP, in order to use the ligation adapters, we need to use a combination of specific restriction enzymes that we have a prior idea about where they'll be cutting, and it is always used in a combination of frequent and rare cutters. Frequent is something that cuts after multiple sites, whereas okay, did I get like a okay, I think I have a question. Okay, I'll come back to the question in a minute. Yeah, in the case of AFLP, we need to use a frequent cutter and a rare cutter. A rare cutter is something that cuts at long distances in a larger sequence, and a frequent cutter is something that cuts after very few sequences. In case of RAPD, we don't need any restriction enzyme at all. And here we are just using two primers in comparison to RAPD, which uses multiple primers. So RFLP, in comparison to both these techniques, AFLP is superior, combining the advantages of both. Let me quickly go to the question before I get back. Okay, is qPCR of functional genes used for determining microbial community composition, or just DGGE and amplified ribosomal DNA restriction analysis used for the purpose? Functional genes. Okay. And the second question is, are operational taxonomic units considered as alpha diversity parameter or just species richness and abundance? Okay, so for question number two, I will be talking about that in a minute. But for the first one, is qPCR functional genes used for determining microbial community composition, or just DG and amplified ribosomal DNA restriction analysis used for the purpose? So those are completely different techniques. So I'm not exactly sure what your question means here, because to determine the microbial community composition using qPCR of functional genes, is qPCR is like mostly used for looking at expression analysis. So if you're looking at functional genes and their expression, you and the composition of these different enzymes in each of these microbial communities, you can use qPCR. Whereas DGGE or amplified ribosomal DNA using restriction analysis can be broadly used for taxonomic classification. That might not give you a good idea about the quantity or a quantitative expression profile, whereas qPCR can give that information about how much of the particular gene is expressed, how much the final protein product is expressed, and so on, like how many transcripts are being made, or all of those information can be obtained from the qPCR analysis. Whereas DGG and amplified ribosomal DNA restriction analysis will only give us like a qualitative information of if this particular gene of this particular allele is present or not. If the answer is still not clear, please do ask again, Prarthana. Yeah, for your second question, I'll come back to you because that's what is going to be our next topic that we are going to discuss. Any other questions in the meanwhile, or shall I continue with? All right, people join like in the last 10 minutes or so. Okay. Uh, Let me just share screen again. Please do confirm if the screen is visible. The screen still visible, right?
Yes, ma'am.
Yeah, thank you. Yes. So this is what Prarthana's question is about, microbial diversity parameters. So we read about a lot of things like species diversity, species abundance, and so on. But how do we numerically represent these things? And why are we talking about this now? Because all the sequence information that we are getting, we need to put that together in a mathematical form so that we'll be able to understand what we are looking at. Like we'll have loads and loads of data from NGS, which we will be seeing in another couple of minutes. Okay. Yeah. Yeah, we'll be seeing in a couple of minutes. So if you have that much data, how do you actually classify it? So, operational taxonomic unit could be anything. It could be a species, it could be genus, it could be anything that we are looking at. So, in a particular environmental sample, you could be looking at the number of species of a particular microbial organism or different genus, or you can use any level of classification for that. And based on this, we can calculate the species abundance and richness. So let's say there's a landscape AB and a landscape CD. So in landscape AB, there's site A and site B. Same way in landscape CD, there's site C and site D. So now the comparison can be done across different levels. So let's say we are looking at one site itself. Let's say we will look at site C alone. Here, whatever is the diversity of microbes, that is called as the alpha diversity. So site C, let's now count the different types of or different colors that are there and different types that are there. So here we can see that this green pattern is there twice, and this purplish pattern, and this orange, and this red. So there are four different types. Let's say these are four different types of microbes. So this is alpha diversity. So the alpha diversity indicates the abundance or the different types of species that are present. It does not indicate the abundance of a particular species. So here alpha value is 4. However, if we look at this particular, let's say all of these are species, this particular species, there are two numbers of that, and this particular one, there are two numbers, whereas these two are just one individuals. So this total number of each of these individual units gives the species abundance, whereas alpha diversity just gives us an idea about the species richness of that particular site. Similarly, if we compare two sites like across site A and site B, that diversity is called as beta diversity. But actually, if we look at this particular image, between site A and site B, everything that is there in site A is also there in site B. So how do we calculate beta diversity? Whatever that is unique to site A and whatever that is unique to site B, and whatever that is shared between site A and site B, calculated only once, is taken. So this is just like A intersection B or A union B that we do mathematically. So everything that is there in these two sites that is unique, as well as something that is present in both the regions taken once, will give us beta diversity. In this particular example, the beta diversity of both site A and site B is 0 because they have everything very similar. And what is the gamma diversity? So gamma diversity is between completely two landscape. Let's say this is one environmental site and this is another environmental landscape, and if you're combining the two and checking what is the diversity, so that gives us an idea about the gamma diversity. So here we'll count the total different types of species that are present, and here these different types, if we count, there are total 6 different types of these species that are present, and hence the gamma diversity here is 6. So this could be okay. Yeah, so this could be either alpha, beta, or gamma diversity, and it could either be species or genus, and it can give us an idea about both abundance and richness. Before I go back to sequencing, are operational taxonomic units considered alpha diversity parameters or just species richness or abundance? I'm not really getting your question, Prarthana, but I don't know if based on my explanation, is it clear for you, whatever that you have asked? If it's still not clear, please do type it out. I can try explaining it again. All right. Since we have only 15 minutes, I want to quickly go over the rest. The next thing is, so where does all this information come from? That is what I was telling. Next generation sequencing actually gives us loads and loads of data in a single time. Initially, when we started off with Sanger sequencing method, only 10 kilo base pairs of data could be generated per machine per day. This was in the early 1980s. But with the development, we invented PCR, later on came microarrays, and then came capillary electrophoresis, and then came short read next generation sequencing. Now we have like single molecule sequencing, like we can even sequence one particular transcript from one single cell and so on, like, and we can go up to kilobase pairs of much higher numbers that have exponentially increased since the development of next generation sequencing. So whatever that we are currently having, that helps us in sequencing, is the third generation sequencing data. So when we have such loads and loads of information, these different techniques like restriction enzyme based technique, PCR based technique, and even the mathematical classifying techniques will help us in simplifying the data and just looking at the data of interest. So this is a brief of how Sanger sequencing and Illumina sequencing works. In case of maybe I think since we don't have a lot of time today, maybe I can go over the Sanger sequencing and the Illumina sequencing in some other class in detail, probably in a section where there's not much to talk about. I can probably talk about these two differences there and explain these two methods in detail. But what we need to take home is that next generation sequencing data can be used for mRNA samples to understand the transcriptome. So whatever I've been speaking about is not just for genomic DNA, it could also be done for RNA, where we get the mRNA and convert it into complementary DNA or cDNA and perform all of these techniques on cDNA as well. So all this information can be obtained for transcriptomic profile of a particular strain or for a whole community, which will be called as meta transcriptome. So this is the history of genomic sequencing, how we started off. So this whole genomic sequencing idea started when the structure of DNA was discovered by Watson and Crick in 1953. Later on, Sanger developed the Sanger sequencing technology, and then came Carey Mullis, who developed the PCR, the polymerase chain reaction technology. And the Human Genome Project was initiated with the development of pyrosequencing. It started in 1990s and it went on till 2003, when the first human genome was completely sequenced. So to get one individual's genome sequence, it took us so many years. But now if anyone wants to get their whole genome sequence, it just takes a couple of days for the sequencing companies to get the whole data and even to analyze the data. If you want to look at, you know, your genome and see if there are some genetic diseases that you carry, something that you need to specifically take care of in your old age. People particularly do this in western countries, where there is more obesity related problems and so on. They do this genetic sequencing so that they can take specific treatments. They start taking these treatments especially for cancer and all that at a much earlier stage before it becomes a problem in the future. So now we have with 454 sequencing, we have sequenced the whole genomes of multiple organisms, including an homo species, which is homo neanderthals, which are humans before homo sapiens. We even have a genome of them, like we got that from the fossil remains of their body, and we were able to reconstruct their whole genome. There are now studies happening where people are trying to replicate a homo neanderthal, to reproduce a homo neanderthal, to bring them back, those early man like people. We also have the genome sequencing done for several HIV viruses and bacteria that cause a lot of infections in humans and so on. So with the help of next generation sequencing, we've been able to progress so far to understand much about much about the genetic information and transcriptomic information of several, not just microbes, but for higher organisms as well. So, what all can next generation sequencing do? It can help us in establishing phylogenetic relationships, the taxonomic position of various organisms. It can give us presence or absence data, whether a particular gene is present, whether a particular gene is absent, whether this particular expression will happen or not, and so on. We can get information about species richness, species abundance, genetic diversity, and also beta diversity pattern between different regions or between different landscapes and so on. So with the next generation data, we can play around so much to get lots and lots of information about different things, starting from taxonomic position to expression profiles and so on. So that's mostly about it for today. We still have like 8 minutes remaining, but I want to keep that for questions if anyone has. Okay, so only species richness and abundance is considered as alpha diversity parameters. Yes, yeah. So for a given, because alpha diversity is only for that particular site, it depends on what we are comparing. If we are comparing across different sites, then we will look at gamma diversity and so on. Then we will look at the gamma. So basically, the alpha, beta, gamma diversity gives information about the species richness and not the abundance. But it can be for a particular site or for two sites that we are looking at, or for completely two different landscapes. It depends on which scale we are comparing them. But abundance has to be looked at a particular site level to see how much of abundance of a particular species is there. I hope that is clear. Based on the explanation, looks like only species richness is considered as alpha diversity parameters because it looks on species diversity and not on. Yeah, that's what I was also trying to get at. Yeah, that's absolutely right, Hitesh. Yeah. Thanks, Prarthana, for bringing questions. So I definitely do encourage everyone to ask questions because that is what will help us in having more discussions, expanding on horizon of understanding these different things. Because not everyone comes from the same background, so it's obviously good to discuss more. We still have like 7 minutes left. I can get into
Hello,
it.
ma'am.
Yes, yeah. Hi, home Manish.
Ma'am, I'm having one doubt from sixth break
Okay,
from the topic that is quantification of microbial biomasses.
okay,
So we can quantify by using several methods such as physical, chemical, and biological. So there is one another parameter that is physiological measurement
okay,
and physical measurement. So what is difference between those two parameters?
okay,
Physical measurement and physiological measurement.
okay. Okay, so physical measurement is anything that is like size and weight and length and so on. So if we look at microbes, it's mostly size and length and all that. So anything that are physical quantities that can be measured in terms of micrometers or grams of physical quantities, whereas physiological measurement is something to do with the internal physiological processes itself. Um, Physiology is anything like how they process a particular let's say how they process how they grow under a specific temperature condition, how they respond to a particular biological source or biological resource or energy source, how what substrates they grow on and how their physiological conditions respond to that substrate and so on. So these are the different physiological parameters that has to do with the internal mechanism of the microbe or the organism of interest, whereas physical quantities or quantities that can be measured in numbers using the standard physical measurements. So that's the difference between physical and physiological.
Thank you, ma'am. Got it. And another question is that uh, how operational taxonomic unit is calculated and what actually it is.
Yeah. So operational taxonomic unit, like I explained today also, it can be any unit like a species or a genus. So we calculate this operational taxonomic unit or the abundance using the sorry, not the abundance, the diversity using alpha, beta, and gamma diversity. So it can be the diversity of the species or the genus or at the order level or anything that we are looking at. So OTU, operation taxonomic unit, basically is this alpha, beta, and gamma values for any given site or a landscape of interest. So if you're given a diagram like what I showed here, like this particular, yeah, like this particular diagram, you should be able to calculate the alpha, beta, and gamma values for this for any OTU that we are looking at, let's say species or genus. Is that?
Thank you, ma'am.
Yeah, you're welcome.
Yes, ma'am. Clear.
Okay. Regarding bioremediation process of polluted environment, does it only depend upon the microbes introduced during the process environment or any other factors? It definitely depends on other factors as well, because like what we have been discussing so far, the microbes need certain conditions to grow, so they don't really behave the same way in an environment along with other microbes, because microbes interact among themselves, they interact with other species, they interact with other microbes, they interact even with the abiotic factors and so on. So these factors definitely need to be taken into account before we actually introduce them into the environment. These are all the factors that will actually influence the process of bioremediation. So these things are definitely tested out before a microbe is introduced into the bioremediation process, because whatever the byproduct that it gives itself should not become harmful, or the byproduct should not interact with other organisms and make it harmful for the environment. So it definitely depends on other factors as well. You're welcome. All right, today was a really engaging session. Today we also had a lot of things to discuss. So I still have to talk about what is the difference between Sanger and NGS. Probably I'll keep it for another session where we have a lot of time left. But today definitely was very interesting for me as well, because many people joined and we also had an interactive session. We could discuss some of the questions as well. So please do join back every week. So we still have at least 5 more weeks to go until this course finishes. So please do join back next week around the same time. Bring your questions. We can discuss the questions and discuss more things that are even outside this course as well.
Ma'am, I have a question.
Yes. Yeah.
Ma'am, all the topics and the methods which we have talked about is this: when we have a culture sample of the microbial community, then we can how we segregate and identify all the processes. These are the methods. But what if we reverse the situation? We don't have the sample of the microbial community. What we have is the wastewater sample, and now we have to identify what community could thrive into that environment. So how would we identify that kind of process?
Okay. So, if I got the question right, so if you already have a wastewater sample, it would mean that there would already be some microbes growing in that. But are you asking like a condition where there are no microbes or anything growing? You just know that it's just the composition of the wastewater or something. Is that what you're trying to ask?
It's just like that. We have a sample of wastewater, and now we have to just to clean that. Just for the example, we
Yeah.
now we know that there are certain bacteria or microbes which we can filter out these all the wastewater materials. So how
Yes.
would we arrange so that a particular microbe fits into that condition?
Yes,
So
yes.
that was
Yeah, so that's what we've been trying to look at using these different methods. So those methods, like you said, they look at microbes that are already there in the environmental sample, but that also gives us an information about what kind of substrates they grow on, what kind of processes that they undergo, how they can convert a particular substrate. In some of the previous classes, we have looked at what are all the different organic substrates that can be used by certain microbes for converting and so on. So if we have that information from prior studies to know what different substrates can be used and so on, and if we know the composition of the wastewater, what kind of waste products are there, if it has a lot of nitrogen content, if it has a lot of methanogens and so on, then we will be able to introduce specific bacteria like methanogenic bacteria or nitrifying bacteria to break down those products into something that is less harmful to the environment. So this whatever that initial screening that we are doing for the environmental samples will give us an idea about the metabolic processes that specific microbes can undergo, which can in turn be used in these environmental samples for bioremediation and other processes.
No. Thank you, ma'am.
Yeah, you're welcome. Okay, thank you so much everyone for joining today. We'll meet again next week around the same time, 6 o'clock to 7 o'clock. And these recordings will be uploaded on YouTube. I hope all of you have the links to these recordings. So in case you joined late and you have missed out on the initial parts, please do go back and check those video links on YouTube. Thank you so much for joining today. See you guys next week. Bye.
Thank you.